Today we will study that what the homolog segregation is and on what feature it depends on the meiosis 1. We must understand that there is a fundamental difference between meiosis 1 and mitosis, which is called as a meiosis 2, and that difference is that in meiosis 1, homologs rather than sister chromatid, they separate and then segregate. वो अलहदा होते हैं और फिर जुड़ जाते हैं. This difference depends on three features of meiosis 1 that distinguish it from the mitosis. और वो differences क्या हैं? That is, the three distinguishing features are, first, both sister kinetochores in a homolog must attach stably to the same spindle pole. उनको एक spindle pole जो है उसके साथ attach होना होता है. This type of attachment is normally avoided otherwise, as shown in the diagram below. Second is that the crossover, it generates a strong physical linkage. एक बड़ा linkage जो है crossover का वो create होता है between homologs, which allowing their orientation at the equator of the spindle. जो spindle है उसके equator के ऊपर वो उसको लगा देता है. Then, as shown that we can see in this diagram, that there is the chromatophores, they are separated, cohesion forces, they are removed, and then the chromatid, they separate all together and segregate in the haploid daughter cells. Similarly, in the mitosis, what happened? That as from the spindle, it removes, cohesion between them is removed, and the chromosomes, they are just separated together, and again, two diploid sister nuclei, they are produced. The third cohesion is removed in anaphase 1 only from the chromosome arms and not from the regions near the centromere. ये बड़ी important चीज़ है. We must remember that the cohesion is removed only from the centromere and not from the centromere arms and not from the region near centromeres. इसी वजह से जो chromosomes हैं वो separate हो जाते हैं, but they are joined with the centromere. This triggers homolog separation in the onset of anaphase 1. जब anaphase 1 शुरू होता है, start होता है, तो ये जो process है ये उसको trigger कर देता है. Now, cohesions near the centromeres are protected from separase. There is an enzyme separase which is a cysteine protease in meiosis 1, and that is protected by a kinetochrome associated protein which is called shigloshin, from the Japanese word of a guardian spirit, because this enzyme guards them from the separase and does not allow them to be separated.